Welcome to BioStudy. In this video, we will discuss about enzymes. Enzymes are biological catalysts that speed up chemical reaction in the living cells without being used in the process. The chemical reaction that a cell or larger living things carries is called metabolism. Many activities in living things are controlled by metabolic pathway which are facilitated by enzyme. Enzymes are basically proteins and their function is determined by their complex structure. And now we will discuss that how enzyme work. Enzyme actually work in two hypotheses. The one is log and key hypothesis. The second one is induced fit hypothesis. And now we will elaborate these two hypotheses. First of all, we will explain that how log and key hypothesis work. This hypothesis was first introduced by Fisher in 1890s century, suggests that each enzyme had a particular shape into which the substrate fixed exactly. This was known as the log and key hypothesis. According to this hypothesis, the substrate is imagined like a log while the enzyme is imagined like a key, as one specific key can open only a specific lock. Similarly, a specific enzyme can break up only one specific substrate. The active site is regarded as a rigid structure that does not modify or change during the reaction process. However, later study did not support this hypothesis in all types of reaction, and therefore the hypothesis was modified into induced fit hypothesis. The second hypothesis of enzyme is induced fit hypothesis. This was first introduced by Koechlin in 1950s. It indicates continuous change in the shape of an enzyme in the response to substrate binding. It means that enzyme can change their shape according to the substrate shape. And these are some substrate and enzyme which take place in metabolic reaction.